you. So, so our second speaker is Dr. K. Kaleshwar Lal, uh, and on behalf of Mahatma Gandhi University and School of Biosciences, I welcome you, sir, to this webinar. And he's a person with expertise in diverse areas, and actually he had his PhD in biotechnology from Kalasalingam Academy of Research and Education. And also, before his PhD, he completed his MTech in biotechnology and also BTech in industrial biotechnology. After completing his PhD, he worked as a postdoctoral fellow in Ariel University, Israel, then as a postdoctoral fellow in Konkuk University, South Korea, then as a postdoctoral fellow at a University of Pittsburgh, USA, and currently he is continuing his research as a fellow at Jeonbuk National University, South Korea. And he is a person with experience in a range of research areas, and he could make very interesting and impressive contributions in his research. In the sense, he is having a large number of publications, more than 41 publications he have, and all these publications, these are in high impact journals, and especially he is having he is having H index of 29. That is very important. So we are very much fortunate enough to have his talk, and even he agreed to help us by providing his talk within a very short time, and he was so kind enough to support us. And with these words, I request Kalesirulal Kalimuthu to continue his talk, and also I suggest students to make use of this opportunity by interacting him effectively, because he can share research experience with you in different countries under different area, and that will be it will be a great opportunity for you. So with these words, I kindly request our resource person, Kalesarwal Kalimuthu, to continue his session. Thank you, sir.
Thank you. Then let's start a basic introduction about selenium and what is a selenium deficiency. In this slide, I am focus only introduction about selenium and selenium deficiency. Selenium is an essential element in our human body and acts as an antioxidative property in our human cells from selenoprotein and selenoenzyme. What is selenium deficiency means if the low level of blood plasma selenium less than 60 microgram per liter, that is called selenium deficiency. In all over the world, the selenium deficiency was affected 0.5 to 1 billion people due to the food intake of selenium differ in various parts of the world. In mainly agriculture soil containing low level of selenium present. So these are the selenium deficiency may cause so many disease. Like I said early, today I'm going to focus only cardiomyopathy and cancer risk. In recent day, so many scientists established if selenium deficiency may cause increasing risk of viral infection, particularly COVID-19. And then if it is overnutrition also may cause so many disease complication. There are three selenium present in our human blood. One is deficiency, acquired, and excessive. As per the WHO report, the dietary intake of selenium for male 75 microgram per day, female 60 microgram per day. There is so many complication occur in deficiency and excessive. So then how much we need to optimum concentration range around 125 microgram per liter, that is safe for our human cells and human body. Then move on to next slide. Um, then move on to protein. I'm more interested in selenoprotein. Currently, there are more than 25 selenoprotein available. Majority which have been identified in a functional group. Some of the selenoprotein does not have a unknown function. Still, research ongoing in this particular field. So, selenoprotein is a protein that includes selenocysteine amino acid in the particular region of amino acid. Among functional characteristics of selenoprotein are five different glutathione peroxidase and three thioreductoreductase, which containing only one selenocysteine. What is the difference between selenium and selenocysteine means? This is the structure of cysteine. This is a selenocysteine structure. Only different is sulfur replaced to selenium. This is a more complication process to translation process. So, among these many member of GPX family, today I am going to focus only GPX4 and GPX1. GPX4 recently in 2012, Stockwell Group published. It's a main regulatory pathway for incubating the lipid peroxidation. In more detail, I will explain in later slide. Next slide. Then today, the mechanism of selenium deficiency to cardiomyopathy, as I said earlier, the low level of blood plasma selenium, it lead to decreasing glutathione peroxidase, it further lead to increasing oxidative stress, finally cause cardiomyopathy. Cardiomyopathy means it's cell death occurred in the cart region. This work was published in New England Journal of Medicine in 2014. Next slide, please. Next slide. Yeah. Now we are discussing how GPX work. I will show the schematic diagram. I hope everyone understand. In this GPX one of the selenoprotein which need selenium from diet. Selenium is integrated with this enzyme and it has been function. Again, it is recognized from the free radical from our body produce and need to remove before any damage. So if a free radical come alone and broken, next slide. Uh, Free radical come alone, it's broken into small piece. Next slide. You can see it's bind with the GPX1 and next slide. It's a the free radical broken and neutralized function in the human cells. So, what I am trying to say is next slide. If the low level of GPX means when the selenium deficiency occurred, if increasing amount of selenium which lead to increasing GPX activity and the product from free radical. If it is increasing amount of selenium intake, 70% activity occurred. When the selenium deficiency patient, if you add more amount of selenium, which increasing the GPX activity. Then I move on to I talk about our regional state of selenium correlation between human cardiac vascular disease in Tamil Nadu population. Next slide. This work was collaborated with Madurai Meenakshi Mission Hospital and approved by Human Ethical Committee. Total 60 patients from different area of Tamil Nadu, South India were studied. Among 30 controlled 30 cardiovascular disease. With the help of doctor, we collect blood from normal people and cardiac patient. Then we have to analyze so many parameter like cholesterol. Next slide. Uh, so many parameter like cholesterol and HRP, protein and selenium concentration measurement using ICP-MS method. The all parameter uh, we analyze this from commercial lab Thyrocare India. Then next slide. These are the result I got from Thyrocare India. There is no significant difference in lipid profile in control and cardiac patient. Then how we have to differentiate in cardiac patient and control based on the HRP is a sensitive marker for inflammation and to predict the cardiovascular disease. Then this serum concentration of HRP is elevated in cancer patient and compared to control. Based on this marker, we confirm that which one is control, which one is a cardiac patient. And then we analyze the selenium concentration also and the glutathione peroxidase assay. In the next slide, I will explain. You can see this graph in control, the huge amount of GPX protein expressed and the selenium amount will be increased. If it is low amount of selenium means the low amount of glutathione peroxide is production which with the help of selenium. So the finally the significant difference were observed in cardiac patient and control. Then this result may help selenium supplement to improve the cardiac health to prevent the free radicals. Next slide. Next slide. So next I am going to talk about the nanotechnology field. Nanotechnology can able to synthesize a material with the specific biological activity in cardiac tissue engineering. That's why I developed the selenium nanoparticles. Next slide, I am going to explain what are the mechanism of selenium nanoparticle synthesis, how synthesis these nanoparticles. When the protein was exposed to 121 degrees centigrade heat, the structure of the buoyant serum albumin will be facilitate and breaking of disulfur bond and further promote to unfold structure of the protein and exposed to more number of SH group. In unfold structure of the SH group could be used as a reduction of selenium ion to selenium nanoparticles, and then based on the color difference we have to confirm that uh, BSA mediated selenium, the A photograph is a red color, in the D is a control group. Based on the color change, we have to confirm due to the excitation of surface plasma resonance, the formation of reaction mixture confirmed by selenium nanoparticle present in this sample. However, but interesting study note that when the same reaction was carried out, some other enzyme like lipase and protease, as you can see this photograph, B and C, lipase protease, they could not produce any color change in the reaction mixture due to the lack of free and exposed SH group present in the sequence, which could not be to contribute the production of selenium nanoparticles. Finally, we conclude that the disulfur bond is a major role for the reduction of selenium into selenium nanoparticles. Next slide, please. The successful synthesis of selenium nanoparticle use to characterize to so many technique like scanning electron microscope, which confirmed by morphology and size. As you can see, the first scanning electron microscope picture synthesis from BSA. The another one is keratin mediated synthesis, which is confirmed by EDX spectra. The red color spot, you can see the figure. The red color spot indicate the presence of selenium element observed. The green color spot, the sulfur element observed by using EDX spectra. Next slide, please. Then we analyze the particle size distribution using dynamic light scattering. Based on the technique, we confirm there is two different sizes of nanoparticle synthesis from BSA and keratin. Next slide. Whether this nanoparticle is more stable in water, we confirm that zeta potential. Based on this result, we confirm our nanoparticle is more stable in water. Next slide. Then we characterize another study, the XRD, X-ray diffraction spectroscopy. This study only which one is this is amorphous structure or crystalline nature structure we need to study it. Based on the previous data JCPD file number, we correlate with our result. It is a more match with the previous result. So this nanoparticles is more in crystalline nature. Then next slide. Based on this conclusion, there are two different nanoparticle size. One is 500, another one is 100. Keratin mediated synthesis and BSA mediated synthesis also. Next slide. Next. Then we move on to toxicity study. Treatment of selenium nanoparticle could be used as a therapeutical agent in cardiac-related disorder, but pre-evaluation of toxicity in selenium nanoparticle is necessary before considering the therapeutical option. To address this issue, zebrafish embryo were selected as an experimental model for evaluating cardiac toxicity. Next slide. Why we are preferring zebrafish as a usage toxicity study? Because this zebrafish embryo is more similar to human, almost 70-85% homology genome level, and it has easy to breed and maintain in the lab. In this embryo is more transparent, so easy to watch heartbeat and development. If it is without any dissection, you can see the toxicity assess visual the heartbeat development in the zebrafish embryo. Because of this advantage, we have to select as experimental model for uh, cardiotoxicity effect in zebrafish embryo. And another one advantage is that there is no need for ethical clearance for zebrafish embryo studied. So next slide. So this slide shows that effect of selenium nanoparticles on zebrafish embryo. Uh, the selenium nanoparticle exposed to 5 different concentration of selenium in the zebrafish embryo. As you can see, this photograph A, there is a control. There is no black color spot in the outer surface of the chorion. When you see the B and the F, you can see the outer surface of the chorion is a small black color spot appear. So concentration dependent selenium nanoparticle observed on the outer surface of the chorion. So we confirm that selenium nanoparticle uptake in zebrafish embryo stage. Then move on to viability of the zebrafish embryo at uh, 96-hour post-fertilization hour. We analyze the low concentration of selenium nanoparticle. There is no mortality were observed. If a high concentration of selenium nanoparticle, significantly more number of mortality were observed compared to the control group. You can see the graph. You can see the different between the p-value. Then move on to next slide. The embryo enter into the larvae stage. This is the larvae stage of zebrafish larvae stage. In this stage, we observed abnormality of pericardial edema and a tail malformation in the zebrafish larvae stage. As I said previously, the low concentration did not show any mortality. So there is no malfunction were observed in the low concentration, such as 5 microgram and 10 microgram. If a high concentration of selenium nanoparticle treatment, there is both pericardial edema and tail malformation were occurred. Tail malformation means it's a curved shape and uh, short of tail. You can see this photograph. You can differentiate between control and tail malformation. Then next slide. Here you can visibly see the difference between the control and selenium nanoparticle high concentration selenium nanoparticle treatment. The circle region is a heart region. You can see the control, there is no inflammation occurred. When the treatment of selenium nanoparticle, the huge inflammation occurred in the zebrafish larvae stage. So similarly, we observed the heartbeat of the high concentration of selenium nanoparticle treatment. So, so decreasing heart rate were observed in the zebrafish larvae stage at high concentration. So based on this study, we found that therapeutic concentration for further study. We used 5 to 10 microgram per ml could be used as a therapeutic concentration for further study. Next slide. So these are we develop one novel biocompatible chitosan selenium nanoparticle film with the help of electrical conductivity for cardiac tissue engineering application. We used H9C2 cell line. We seeded on the top of the film. After 24 hours, the cells are grown well on this film, and then we characterize so many technique uh, to confirm that selenium, this film, it's a more biocompatible and easy to implant to cardiac tissue engineering in vitro study we confirm. Later on, we want to study more about in vivo study. Um, then move on to next slide. Uh, this is a more important topic I'm going to share with you all. Cancer is a multifactorial disease, causes 9.6 million people death in annually. So today I'm going to discuss about only triple negative breast cancer. What is triple negative breast cancer means there is no receptor present in the outer surface of the cells. There is no targeted therapy nowadays available. So that patient is more poor prognosis. Another one interesting study, almost four time higher selenium level in the breast cancer compared to healthy surrounding tissue that indicating the redistribution of selenium because selenium, one of the enzyme selenocysteine lyase, this enzyme is redistribute to protect our cells. That is one of the enzyme maybe involved uh, in this breast cancer. So we need to study more about that enzyme later on. So because of selenium present, no, no, no. So selenium present means if any free radical come like chemotherapy to generate ROS to prevent from selenoprotein and selenoenzyme. That's why chemoresistant is a main challenge in the triple negative disease. Nowadays, there is no drug available for clinical trial. Uh, Next slide. Uh, so overall, we have to uh, alternative pathway of triple negative breast cancer. We need to talk about here. One mechanism recently found that in 2012, the Stockwell group developed ferroptosis. The ferroptosis is more important for survival of triple negative cells and is closely related with glutathione peroxidase 4. This ferroptosis pathway is a non-apoptotic formation of cell death characterized by iron-dependent lipid peroxidation. As you can see, there is a pathway. When the glutamate cysteine dipolar reaction, which lead to glutathione production and which further form GPX4, it's to lead to lipid peroxidation to protect the lipid peroxidation. The simultaneously, the other ion pathway is the main role involved in the ferroptosis. If it is inhibit GPX4, the ion pathway on to form the Fenton reaction to form the lipid peroxidation to further lead to ferroptosis. Is the main role is GPX4 is a regulator for this cell death pathway. Next slide. So the selenium involved in a cancer study when the triple negative breast cancer cell line were cultured in the new physiological medium is a more interesting study published in Science Advance. Uh, they develop a new medium like Plasmax. That is one of the selenium medium. They analyze two different kind of medium. One is selenium medium, another one is conventional medium. All of you know about DMEM F12. These two different media they cultured and grow in colony formation assay. As you can see, this figure B, there is concentration of dependent selenium added with DMEM. The triple negative cancer cell gradually increasing the colony formation. The large amount of colony formation in 10 nanomole TMEM. Then they analyze two different culture. One is DMEM F12, another one is DMEM F12 plus sodium selenate. Only different is there is a seeding different, only seeding different in all the plate. The first plate 500 seeding cells, the last plate for 40,000 seeding cell. So you can see the different, the first two plate, the huge number of colony formation occurred in this plate. There is no significant difference in the high density cell seeding. Similarly, they check whether GPX work or not. So in this Western blot, you can see the different low density and high sensitivity also. The GPX4 is a main role to protect the cell from lipid peroxidation and ferroptosis. So finally, selenium is a main role to prevent the uh, cancer cells, particularly triple negative breast cancer cells. Next slide. So another one study, several study suggest that FAN, fatty acid synthase, is a main molecular target for triple negative. So one of the study, the high selenium treated, the more number of fatty acid synthase. So in my point of view, the selenium involved in different pathway like lipid metabolism, glucose metabolism. There is interconnecting. There is a crosstalk between the three different pathway. We need to study more about particular pathway. We need to find out particular mechanism. Next slide. In recently, Geological Society of America published one novel method, uh, which deep ecosystem, how it is survey in the deep ecosystem, which energy is produced from the microbial survival in the deep ecosystem. They found that selenium is a major role involved in the reduction and oxidation process. If it is a small amount of selenium present in this deep biological ecosystem, it may be microbe will be survival. In my hypothesis, in tumor microenvironment, there is small amount of selenium present means the cells will be survival easily to grow faster. Particularly the triple negative cancer cell is one of the cancer stem cells. So my hypothesis, selenium, we need to remove detoxification from the cancer tissue. Next slide. In future, there are so many paper published in previously different stage, different biomarker, tumor marker like KRAS, HER, but I am not believe that marker, because every time to change the surface protein or surface receptor and human biomarker change every time. So nowadays the people, scientists, is there any trace element suitable biomarker for diagnostic purpose, both cancer and cardiomyopathy? This work recently focused on German people, German scientists. So I am also think that this research also want to contribute in the future development of test measuring selenium status directly in the one drop of finger. It should be possible at home, residence care, and during the cancer treatment. So everything related selenium, selenoprotein is more helpful our human body and more danger to in our human body also. We need to check day-to-day life how much amount of selenium we intake in our human body. We need to check like glucose meter. We check every time, right? That's we want to check in future study. Thank you, one and all. I hope everything understand my talk. Thank you very much. Give me the wonderful opportunities. Thank you very much.